Good afternoon. Want to welcome everyone here on Penn campus, those that are visiting and others congregating for Jake Hoffman's presentation for his thesis. I do want to also just check. I think we have people calling in remotely. So you could put in chat or let us know. I don't know how you know if you can't hear me, but try to get a message here if you can't hear us. And Jake, we will try to talk into the microphone. So I ran into Jake outside as he was getting ready to come in. And first of all, this is about as dapper as I've seen Jake. So it's, let me just say, it's a good look. But he'll not only dazzle you in that way, but I'm sure with the depth and enthusiasm of his science. And so, for those that aren't familiar with the process, this is somewhat of a rite of passage. This is called a thesis defense, and there's a committee that's been overseeing Jake's work from the very beginning, a distinguished faculty, and they're all here. But importantly, the first part of this session would be for all of you to participate in his lecture that summarizes the research that he's been doing, at least some of the research that he's been doing while he's been in my lab. And my name's Jim Wilson. And then a question and answer for those in the audience. And then following that, we'll excuse you and you will go about your day. And Jake will stay here with the committee and answer additional questions. And then the committee will determine whether he passed or failed. We have high expectations that this is going to go well today, but just so that people understand kind of the process. Want to give as much time as we can to Jake. So Jake, I'll pass it over to you.
Thanks very much for coming on, everyone. I appreciate it. My name is Jacob Abraham Hoffman, and I am honored today to present to you my doctoral dissertation titled Modulation of the AV9 Galactose Binding Pocket Yields Novel Gene Therapy Vectors and Predicts Cross-Species Glycan Profiles. And we're going to start our story today with you. You are an organism made up of organs and tissues, made up of cells, and inside each of these cells, you have your genome. This genome is made of DNA, and it is the blueprint for everything that you are. So how does this genome get expressed? We call this the central dogma of molecular biology. And you start with a gene in your DNA that gets transcribed into RNA, that gets translated into a protein. Proteins do so many different things. They allow you to eat, they allow you to move, they allow you to think. And your continued existence right now depends on the coordinated action of thousands and thousands of different proteins. But what happens when something goes wrong? What happens if you have a mistake in your genome, a mutation in your DNA? That mistake gets transcribed into RNA, and that gets translated into a protein that may not work the way it's supposed to. If one of your proteins isn't working the way it's supposed to, you can get very sick, and we call this a genetic disease. So there are about 7,000 diseases with a known causative gene mutation. Some examples are inherited retinal disease, spinal muscular atrophy, and hemophilia. And most of these diseases have no or poor treatment options. So what we do in the Wilson lab is gene therapy to reverse this disease phenotype. So here's what that looks like. We take this faulty copy of the DNA, we're going to move it to the side, and instead we're going to insert a healthy copy of this gene. That healthy copy gets transcribed and translated into a protein that will hopefully reverse that disease phenotype. But how do we do this? How do we get a healthy gene into the cell? And that brings us to the hero of our story, the adeno-associated virus, or AAV. AAV is of the genus Dependo parvovirus, Dependo meaning to be attached to as a condition or a cause, and parvo meaning small. AAVs were discovered on accident. So in 1965 at the University of Pittsburgh, we had some researchers and they were looking at adenovirus, which is this thing right here, under a microscope. what they were surprised to find was all these smaller viral particles associated with that adenovirus, hence the name adeno-associated virus. They tried to culture these AAVs on their own and found that they couldn't reproduce without the help of some helper virus such as adenovirus. It turns out that AAVs have outsourced a lot of their replication machinery to these helper viruses, which makes the AAV genome very small. So this is the adenovirus genome. You don't need to know anything about it other than the fact that it's big. It's about 36 kilobases. And now we can contrast that to the AAV genome, which at 4.7 kilobases is much smaller. AAV has two primary genes. The first one here is called rep. That rep gene is involved in replication and packaging. There are also a couple accessory proteins, AAP and MAAP. But then the gene we care about the most today is this one here, CAP. This CAP gene codes for the viral capsid proteins VP1, VP2, and VP3. And these three proteins combine to make the viral capsid. This capsid is the business end of the virus. It's what allows it to get inside of a cell and do all the fun gene therapy things we want to do. Now, the last portion of the AAV genome that we want to talk about are these things here called the ITRs, or inverted terminal repeats. ITRs have a number of different functions, but what we care about today is that they act as the packaging sequence for this AAV. So anything that is flanked by these ITRs will get packaged into the AAV capsid, creating a complete adeno-associated virus. Now, there are a bunch of different kinds of these AAVs. There are serotypes 1 through 9. Each one has a different capsid gene, and each capsid gene creates a different capsid protein, and all of these AAVs are going to have different properties when we try to use them for gene therapy. But we don't care about adeno-associated viruses. We care about AAV vectors. So what's the difference? So we're going to start with our AAV genome to make a vector. And the first thing we're going to do is we're going to hollow it out. We're going to take rep and we're going to take cap. We're going to move them over here to this thing called the transplasmid. This transplasmid contains all of the AAV genes necessary to make an AAV vector. But it does not contain those ITRs, so it can't be packaged into the final vector. Remember all these different AAV serotypes that we had, 1 through 9? We can take that cap gene from any of these serotypes and clone it into this transplasmid, which gives us modularity and allows us to choose which capsid we're going to use to do gene therapy. Now, we're left with this hollowed out AAV genome. We've got our ITRs, and in between it, we can put what's called a transgene. This transgene can be anything that we want it to be. It could be the healthy copy of a protein that could cure a genetic disease, or it could be something we call a reporter gene, such as GFP. And this allows us to see where that vector is going. So we take this transgene and these ITRs, put them on what we call a cisplasmid, and anything that is flanked by ITRs on that cisplasmid goes inside of the vector. So now we have to come back to our adenovirus genome, steal a couple genes that are required for AAV replication, and we call that a helper plasmid, and then we move to AAV production. AAV production happens inside of the cell, and we take the cisplasmid, the transplasmid, and the helper plasmid and transfect them in. Off of that transplasmid, we get expression of the capsid proteins, and off of that cisplasmid, we get replication of the AAV genome containing our transgene. This transgene then gets packaged inside of our AAV. Creates an AAV vector. Coming back here, we can now use our AAV vector to deliver that healthy copy of our transgene to be transcribed and translated into a protein that, if we've done our jobs correctly, can cure a disease. This process is called transduction, and it is the limiting factor for gene therapy. To think about when we're doing gene therapy is this concept of biodistribution, or where a vector goes after it's injected. So here we have an organism. This organism has a heart and it has a liver. And we're trying to understand when we take an AAV vector and inject it into this organism's bloodstream, where does it end up on this graph? So some vectors will end up in the liver. Some vectors won't work very well at all. They won't go to the heart or the liver. Some vectors will go to the liver, and other vectors will even go to the liver. It turns out of the natural AAV serotypes, there aren't a lot of vectors that are really good at transducing the heart. Now, if we want to do gene therapy in the heart, there are a couple ways around this. You can increase the dose of your AAV, and that'll absolutely increase the amount of cardiac transduction that you're seeing. However, you also increase the amount of liver transduction that you're seeing. And when you get to this portion of the graph, you run into safety concerns. These safety concerns can be very serious. There are a number of instances of patients who have died or gotten seriously ill after receiving a high-dose gene therapy. And this area is something that our lab in particular has put a lot of work and effort into characterizing and understanding in an effort to make these gene therapies safe for the patients who need them. Another problem with high-dose AAV gene therapy is that it's expensive. So these are the 4 FDA-approved gene therapies currently on the market. For a single dose of these, you're running anywhere from $825,000 to $3.5 million. These therapies are incredibly expensive, and a large part of this is the cost of manufacture. So when even lab scale preps of these AAVs can cost thousands of dollars, anything that we can do to reduce the amount of AAV that we need to use to treat a patient is going to make gene therapy safer and cheaper. Now, if using a low dose is so good, why don't we do it? And the answer is because it doesn't really work. So there have been attempts at treating cardiac diseases with low-dose gene therapy. In 2015, they had these CUPID trials. And unfortunately, these trials failed in phase 2. They weren't able to correct the disease phenotype. And this failure in kind of a post hoc analysis was found to be due to a lack of transduction. There's thus an unmet clinical need for these liver detargeted cardiac transducing vectors. And it's with this idea in mind that I started on my PhD. So let's review. We've got transduction. You've got your AAV, it enters into the cell, it puts its genome inside, and then we get expression of the transgene product off of that gene. This is an oversimplification. Turns out it actually looks a bit like this. And any one of these steps in this process, a PhD student could spend their entire time studying. I spend my entire time studying this step right here, AAV binding to glycosylated cell surface receptors. So let's break that down. AAVs bind to cell surface receptors. So these receptors, they're just proteins, but they're proteins that live on the cell membrane. So here we are, we have our cell surface receptor, but AAVs bind to glycosylated cell surface receptors. So it turns out this is the central dogma of molecular biology that I've been showing to you guys. And I promise I've only been lying to you a little bit. It turns out there's actually another step called glycosylation, where we have these proteins that then get decorated with these chains of molecules called glycans. So what's a glycan? So there are a number of different kinds of glycans. We're only going to talk about N-glycans, which is a particular class here. And these are chains of sugar molecules that are present on about 70% of proteins. There are 5 sugar subunits: sialic acid, galactose, mannose, fucose, and glycnac. And there are many different glycoforms of this as well. So they can look like this, they can look like this, they can look like this. There are countless different variations of what these can look like. And so we have our glycosylated cell surface receptor, we have our AAV, it can bind, and then through a series of processes that still isn't fully understood, it can get internalized into the cell. What I want to focus on today is this interaction right here between the AAV and the glycan. So remember all these different AAV serotypes. We're going to focus just on this one here, AAV9, and its ability to bind to its cell surface receptor, galactose. So here's the AAV9 capsid. And in red, I've highlighted the galactose binding pocket on this capsid. There are about 60 of them. Now, this galactose binding pocket was originally discovered by a grad student in our lab, Christy Bell, back in 2012. If we zoom in on this binding pocket, we can see that it's made up of 5 amino acids: D271, N272, Y446, N470, and W503. And galactose fits right in there and can bind to it. So the first thing I wanted to do was measure the interaction between AAV9 and its receptor galactose. So we moved to an experiment involving these galactose beads. So these are just beads. They've got galactose coating the outside. And I wanted to see how well AAV9 could bind to these beads. But I also wanted to test its ability to bind to those beads in the presence of increasing amounts of galactose to see what we would see. So here's what that data looks like. On the x-axis, we have free galactose concentration. And on the y-axis, we have genome copies per mg beads. This is how many AAVs are bound to those beads. And so you can see here that at low galactose concentrations, we have a high level of AAV9 binding to the beads. And then as we add more and more galactose in, we compete that binding interaction away down to baseline here. So this is nice, but we need a negative control. So we moved, so another of these variants that Christy Bell identified is this W503A mutant, where you take this residue, W503, and you change it into an alanine. So it becomes W503A, this galactose-deficient binding protein vector. So if we do the same experiment with W503A, you can see we've got this baseline here, that W503A doesn't care how much galactose is in the solution. It's not going to bind to those galactose beads. Okay, so now we've looked at this at the galactose interaction in a very targeted manner. Let's move the next level up and let's look at cells. So we're working with two cell lines here. You've got Pro5 cells on the left and LEC2 cells on the right. Pro5 cells are wild type and they have these complete glycans adorning their surface. On the right, however, these LEC2 lines, they have a mutation that stops them from adding these sialic acids, so all of their galactose residues are exposed. When we test AAV9 on these cells, we see a difference. So here we have, on the y-axis, we have genome copies per well. This is a measure, again, of binding to these cells. And then you can see that for AAV9 on Pro5 cells, you have low levels of binding. And then on LEC2 cells, you have high levels of binding. Then when you add galactose back into the media, you break that ability to bind to these LEC2 cells and it goes down again. We can see this visually by looking at the cells under a microscope. So any cell that this AAV gets into is going to glow green. You can see Pro5 cells, no transduction, LEC2 cells, transduction, LEC2 plus galactose, lowering that transduction. W503A doesn't care at all. W503A can't bind to this galactose. And so on Pro5 or LEC2 cells, regardless of whether galactose is in the media, we don't see any transduction. Finally, we wanted to look at these in vivo to get a really good baseline for what these vectors represent. So we took AAV9 and W503A, we put them into mice for 2 weeks, and then observed liver and cardiac transduction. So in the liver, you can really see this difference. AAV9 lights up the liver. It's very highly transduced, whereas W503A, you can barely see anything. In the heart, it's a little bit different. Here we have AAV9, and AAV9 has okay heart transduction. It doesn't look anything like the liver. But then W503A has this weaker heart transduction in addition to this weaker liver transduction. So AAV9 and W503A, these are our positive and negative controls moving forward. So we need to remember them for the experiments that we're going to do. So now I was wondering, what happens if we do more than just an alanine substitution? What happens if we take W503, Y446, and N470 and make every possible combination of every amino acid in those 3 positions? What I was hoping for is that we'd find this liver-detargeted cardiac vector, but that also comes out to about 7,000 different variants that I'd have to test and absolutely did not have time for them. So we had to move to an AAV library. So what's an AAV library? You'll remember when we're making an AAV vector, we take rep and we take cap and we get rid of them. When you're making an AAV library, however, you only get rid of cap. You only get rid of rep, keep cap around, and then you make all 7,000 different variations that you're going to make to that capsid sequence. Then when we use this to create our vector, we are left with a pool of all 7,000 different AAV capsids. And importantly, each of these capsids contains the genetic sequence for that capsid itself. Another way of thinking about this is that each of our AAVs has a barcode. And if we scan those barcodes in different situations, we can understand which AAVs are present in which settings. So we wanted to test this. This is our experimental setup. We've got our library cap plasmid. We've got Y446, N470, and W503, which comes out to 7,000 possible combinations. And then we gave each variant in this library a 3-letter code. So AAV9 would be Y and W, whereas W503A would be Y and A. This is just how we can understand what we're looking at when we look at any of these vectors. So we took this library, put it in mice at 1E12 GC per mouse, waited 14 days, took heart and liver, extracted RNA for Illumina sequencing, and this is what our data looks like. So on the x-axis here, we have liver enrichment. This is how well our vectors are transducing the liver. And on the y-axis, we have cardiac enrichment, which is how well our vectors are transducing the heart. Each individual point on this graph is going to be an individual vector. So here we have our positive control, AAV9, with strong liver enrichment and okay cardiac enrichment. And then here we have W503A, our negative control. It's not transducing very well either tissue. And here's the rest of our library. So a couple things stand out here that I want to draw your attention to. The first is that we've got this cool looking curve. It starts down here, and in the first section of this curve, we have vectors whose cardiac transduction is increasing faster than their liver transduction. We get to a point, and then liver enrichment starts to dominate. It's almost as if the liver is pulling vector away from the other organs to transduce the liver. The second thing to notice is, hey, we've got a bunch of vectors that look like they are doing better in the heart than AAV9 and doing worse in the liver than AAV9, which is exactly what we want. This is a little early to get excited, though. We're working with 7,000 different vectors. We've never done an experiment like this before, so I wanted to dig a little bit deeper into these results to understand what was going on. So I drew this line. This is the one-to-one line. And we called any vector above this a heart vector, and any vector below this a liver vector. I then looked at each position, Y446, N470, and W503, to understand which amino acids were represented at these positions. So here's what that looks like. On the x-axis, we have all the different amino acids that we're looking at, and on the y-axis, we have the percentage of variance with that amino acid at position 446. And so we don't see any really strong trends here. If we look here at tyrosine, that's the wild-type amino acid. We maybe see some clustering within this amino acid class, this aromatic class, but all these other amino acids, there's no really strong preference for any of them. That's fine. We look at position 470, we see a similar thing, maybe a slight preference for amino acids in this same polar bin, but other amino acids are permissible. And now we can contrast this to position W503, where things get a little bit more interesting. Here we can see that the total number of permissible amino acids is highly constrained. And the vast majority are either the wild type tryptophan or this alternative aromatic amino acid, histidine. In particular, our cardiac vectors seem to favor histidine at this W503 position. So then I went into the literature to do a little bit of reading, and it turns out that glycans actually like to bind to these guys right here, these aromatic amino acids. So these are the results of a study from 2015, and the researchers went in and they looked at every single glycan protein interaction in the protein database and found that of all the amino acids, it's these aromatic amino acids that are the most common to bind to glycans. So great, this makes sense. Glycan binding amino acids are favored at position W503. This makes sense given our experimental design. Let's move forward. So the next step is a more targeted screen. So here we have our library cat plasmid. This time we're taking the top 275 variants. And so this is a smaller, more targeted library. And because it's smaller and more targeted, we can do this fun 3x synonymous codon control for each variant. So what does that mean? The way you can think of this control is by words that sound the same but are spelled different. So our example would be the word color. We also have color or color. They all sound the same, but they're spelled different. And we take that concept to make up each individual vector in our library. So in our library, there are 3 different versions of each data point, which then comes together into one individual data point. And so what you're going to be seeing in the next slide is this big data point right here, but just know that it's supported by all these 3 other ones below it. So took the second round library, same experiment, put it in mice, take them down, see what we see. On the x-axis here, liver enrichment, y-axis cardiac enrichment. And the first thing to notice is that we've recapitulated our curve from the first round library. That's good. We want to see that. That lets us know that we may be onto something. But then the most exciting part of this study was these vectors up here. So right at the top of the cardiac enrichment scale, we have this set of vectors with a shared XRH motif. So you can see that we've got HRH, GRH, FRH, SRH, VRH, TRH, ARH. All of these vectors have the same pattern, and they're clustering in the same area on this graph. Importantly, this is the area of the graph that we wanted our vectors to be in, with high cardiac transduction and detargeting from the liver compared to AAV9. So this was exciting. Decided, hey, let's take 3 of these and test them individually in mice. So we moved into a dose response study. And here we take our 4 vectors, AAV9, and our 3 XRH vectors, and we put them into mice at 3 different doses, 5e12, 1e13, and 5e13 GC per kg, which is a low dose, a middle dose, and a high dose. So we injected them, waited 14 days, and then looked at their hearts and their livers to understand if these vectors are actually better. So because we were being a little bit more intensive about this study, we wanted to look at DNA, RNA, and protein. And so in the liver, at the DNA level, this is what we found. So on the x-axis, we have all of our different vectors. We have a fold change compared to AAV9 here. And on the y-axis, we have GC per diploid genome, which is essentially how many vectors got into each cell. And what we can see is that all of our XRH vectors, and in particular HRH, which ended up being our best vector, is up to like almost 6-fold decrease compared to AAV9 in the liver. This is a significant result. At the RNA level, this is also borne out at the 2 lower doses. We can see that, that HRH, again, this 5 to 6-fold decrease compared to AAV9 in the liver. And we can see this with our eyes too, when we look at GFP expression. So here we have our different vectors. We've got our high dose, our middle dose, and our low dose. And if you compare HRH to AAV9 at any of these doses, you can very clearly see that we're getting less liver transduction. That's awesome. That's exactly what we wanted to see. But now we have to move to the heart. So in the heart, things are a little bit more difficult to parse. So at the DNA level, we actually don't detect a difference between any of these XRH vectors in AAV9, which was a little bit surprising. However, in the liver, once we get to these, at the RNA level, especially once we get to these lower doses, we actually start to see separation between the 2. So you can see down here at the RNA level in our lowest dose, HRH has a 10-fold increase in the heart compared to AAV9. At the protein level, we can also see this. So this is immunohistochemistry. We did staining for GFP. So anything that's brown means it was transduced. And again, comparing AAV9 at these high doses to HRH at these low doses, you can see with your eyes a difference in the amount of cardiac transduction that we're getting. So this is awesome. This is exactly what we wanted to see. The XRH vectors, they work in mice. Next step, let's move to non-human primates. And so I want to take a minute to talk about translation, moving between mouse, non-human primate, and human. And we do this often in science, but there's no guarantee that something that works in a mouse will work in a non-human primate. There's no guarantee that something that works in a non-human primate will work in a human. Additionally, non-human primate studies are very expensive and very time-consuming. And from an ethical standpoint, they are closer to humans, so we want to minimize using these non-human primates as much as possible when we do these studies. So instead of taking the XRH vectors individually into these non-human primates, we instead took that second-round library that we screened earlier in mice, put it into 1 NHP, and did the same experiment 14 days Illumicines. And here's that data. So things look a little bit different here. The first thing you'll notice is that the curve that we saw for our mouse data, that kind of goes away. So this is interesting. It lets us know that, hey, maybe things in non-human primate are a little bit different than they are in the mouse. Surprise, surprise. And we also see that these XRH vectors, they're still performing well in the hearts compared to AAV9, which is down here. However, the amount of liver transduction that we're seeing is things look a little bit different. And here's what I mean by that. So if we take our mouse data and we take our non-human primate data and we line them up, what I want you to look at is AAV9 and these 2 vectors here, FNW and HNW, in the mouse, and then compare them to where they are in the non-human primate. And you can see that they shift left. That was kind of not what I was expecting, especially since most of these XRH vectors and all the other vectors, they're jittering around a little bit, but they're not shifting to the same degree. Another way to look at this is by comparing the RPM of these vectors in mouse liver and in non-human primate liver. So this is essentially looking at the correlation for each of our vectors between these two model organisms. And what you see is that there's this, if we plot a trend line, the slope is very close to 1, which means most of these vectors do about the same in non-human primate liver as they do in mouse liver. The exception is these 3 vectors here, AV9, HNW, and FNW. So the interesting thing about these vectors is that they only differ by 1 amino acid. And that amino acid are these alternative aromatic amino acids, those aromatic amino acids that we were talking about earlier. So this isn't enough to go on yet. We need more information. So let's take a look at galactose binding. So we wanted to test both the library and these individual vectors on their ability to bind to galactose. So the first thing we did is we went back to this galactose bead experiment. But this time we did it with the library. And so here's what that looks like. On the x-axis, we have our galactose concentrations. Here we have no galactose and then increasing amounts of galactose. And then on the y-axis, we have this enrichment value on those beads. When we look at our library at 0 molar galactose with no galactose involved, a couple things stand out. First, we've got our negative control, W503A, it's right down here. We've got our positive control, AAV9, up here. We can see that AAV9 binds to galactose, W503A does not. And then these 2 vectors, HNW and FNW, they are also sticking out in the same way that they were sticking out in the non-human primate liver. Another thing to note is that these XRH vectors here, they don't seem to bind galactose very strongly at all, which is not what I was expecting. Now, when you add increasing levels of galactose, you can see that the difference between these vectors and the rest of the library starts to go away, normalizing once you get a high enough level of soluble galactose in solution. We also tested this individually. So on the y-axis, we have our genome copies per mg beads, and on the x-axis, we have all our different vectors. I've compared all of these guys against AAV9, and what you can see is that HNW and FNW have the highest galactose binding besides AAV9, and they're not statistically significant. They're not significantly different from AAV9. The XRH vectors, W503A and AAV8, on the other hand, except for VRH, which chose not to behave, they do not have these high levels of galactose binding. And then, so we wanted to really drill in on this and make sure that what we're seeing was real. So we tested the library and these individual vectors on Pro5 and LEC2 cells. And this is what we see. So if we remember from earlier, AAV9 has this low transduction in Pro5 and high transduction in LEC2 pattern. We see that HNW and FNW also have that same pattern, whereas SRH, VRH, and HRH do not. W503A is down here. It's kind of just struggling. That's fine. And then we have in LEC2 cells, if we look at just the expression that we're getting, you can clearly see that AV9, HNW, and FNW are their own class of glycan binders, able to transduce these LEC2 cells, while all other vectors are performing more poorly. Finally, when we look at the library, we see the same thing. So on the x-axis, we have RPM in Pro5 cells, and on the y-axis, we have RPM on LEC2 cells. You can see we've got this cluster, which is the majority of the library, and then AV9, FNW, and HNW, again, are sticking apart. So this is interesting. We've got the same set of vectors that is performing weirdly in 2 completely separate situations. The first in these cells in its ability to bind to galactose, and then sticking out due to its inability to transduce the non-human primate liver. So what I wanted to do was do a glycan profiling experiment to understand which of these glycans are expressed in non-human primate tissue, with the hypothesis that maybe NHPs have lower levels of exposed galactose. Now, I wanted to do a glycan experiment, but I didn't know what that was. So I went to ChatGPT and I said, hey, ChatGPT, show me a glycan profile. And what I got was this: glycans exposed, shocking glycan secrets, hidden truths of glycan. So I know this seems silly, but ChatGPT was actually onto something, because the question that I'm trying to answer is which of these glycans are exposed. So, so, let, so let's think of this from the perspective of a vector looking at a cell. You've got all of these glycans that are sticking out, and you've got these terminal exposed residues. Those are the only residues that the AAV can see as it's coming down to the cell to bind to it. And only some of these glycans are going to be galactose exposed. So only some of them are going to be galactose. So what I wanted to do was count up every glycan in a sample, then count up each exposed end on those samples, and then calculate the percentage of exposed glycan ends that are each of the different types of glycan subunits. So in this example, we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 exposed ends. 4 of them are galactose, so this would have 40% galactose exposure. Okay. So then we didn't know how to do this, so I went to the metabolic lab at CHOP and worked with Miao and Idris with their, and they have a clinically validated glycan profiling mass spec pipeline. And so here's what that looks like. So we have our liver sample, and we take it and homogenize it, and we isolate these glycoproteins, right? These proteins that have glycans attached. Then at CHOP, what they do is they take this enzyme called PNGase F. This PNGase F goes and cleaves all those glycans off of our protein. We then take those glycans, analyze them on a mass spec machine, and that gives us something that looks like this. This is a big, scary mass spectrum with a ton of things that are going on. And I was very lucky that Miao and Idris would then take these results and put them into an easily formatted Excel spreadsheet for me to analyze. So the first way I wanted to test this was to validate it and say, hey, if we know that there's a difference in the exposed glycans on these samples, are we going to be able to see it via glycan profile? So here is a list of all the glycans that we looked at in our sample. You absolutely don't have to know all of them. And then we took the CHO-LEC cell lines, so those LEC2 cells and those Pro5 cells and a couple other similar variants, and we tested them using this pipeline. So we've got our different cells, and then so now I'm gone in and color-coded every one of these glycans, red to blue, based on how much of that glycan exists in the sample. So red means there's a lot, blue means there's not very much. So this is what our data looks like. So what we're trying to compare here is how much red or how much blue there is in one of these different columns. So where the differences are really stark are in these LEC1 and LEC8 cells, which can't even add galactose to their glycans. So they're obviously going to have much lower levels of exposed galactose. And you can see LEC2 and Pro5 cells much more red than these guys. Also, if you compare directly LEC2 versus Pro5 cells, it seems like, yes, there are more of these galactose-exposed glycans being expressed in these cells. Now, when we go in and do our calculation and count up all the exposed ends, this is what that looks like. And so for LEC2 cells, we've got a probably like 24, 25% exposure of these glycans, whereas in Pro5 cells, we have about a 20%. We're able to detect a statistically significant difference, which says, hey, okay, we are able to see when there's differences in galactose presentation. So the next thing we wanted to do was look at our samples, look at mouse versus non-human primate liver and see what that looks like. And so here's that data, and here the difference is much more stark. So we can see our mouse liver glycans are very red compared to 2 different species of non-human primates, cyno and rhesus, which have a lot of blue, and so are clearly having lower amounts of these exposed galactose residues. If we do our counting up and calculation of the exposed glycans, we can see that this is borne out in that as well, with a statistically significant reduction in the amount of exposed galactose residues in these non-human primates compared to mouse. Now, I also threw in one more arm to this trial just to see what we would get, and that was human samples. And with human liver samples, we actually found something we weren't expecting, which was that their exposed galactose profiles seem more similar to mouse than they do to non-human primates. And here's what that looks like in when we count everything. So, in conclusion, a couple things. One, what this glycan profiling experiment shows us is that NHP models may be underestimating AAV9 liver transduction in humans. They have different levels of exposed galactose, and we know that galactose is necessary for this liver transduction. So this is something that warrants further study. I want to stress that this is a very, very preliminary result and requires a lot more teasing out to know if it's actually true. One way we could do that is to analyze the transduction patterns and glycan profiles in liver humanized mice. This is a potential future direction for that. Second, we found these XRH vectors that are promising as gene therapy vectors. So they're able to transduce the mouse heart very well. They are detargeted from the liver, and in non-human primates, they do seem to retain some of that cardiac transduction ability. So next steps would be individual testing of these in non-human primates, because there's a lot of work that has to be done before a vector can actually become a potential candidate for gene therapy. Finally, we've also shown that galactose binding is not necessary for transduction of all tissue types. So we have these XRH vectors. They don't bind to galactose, but they're able to get into the cardiac tissue and transduce it. So our next steps here would be to analyze XRH binding to other factors such as AVR or serum. And with that, thank you for coming, and I'd like to open it up for questions. Thank you. So we actually tried to do a library screen on the other two. It turns out, and so we made two different libraries with the first three and also with the last three. And we produced them both, and the first two residues, that library didn't package very well, so we just didn't have enough of it. So we're like, okay, drop it in, and then moved on with the other one. So we would homogenize the tissue, and then we heat and activate it, and then we do the PNGase F digestion, which cleaves the glycans from the proteins, and then you do Hillet chromatography to. Yes. We potentially could. That would require us to change around the process, but I think it's possible. So. So yeah, that's a great question. I actually think that data sets like this, all these vector engineering data sets, they're a very good application for artificial intelligence. The way these models work is you essentially make a giant spreadsheet with like all of these different values for all these different conditions, and then you use that to train an AI model. And what we're generating here is we can have a list of all these different vectors with a number next to them for each tissue. And so, yeah, I think AI could be really useful for analyzing these kinds of data sets. It just requires people with the knowledge of those tools to apply to. I was trying to. That's something I would like to study further in the future. Yeah. Yeah, absolutely. So, I mean, what we're doing, we're saying, hey, we've got this binding site, and we're just going to play around with it. We're going to see if we change the shape of it, is it going to have different properties? And so we can take a similar analysis in really any binding site that we want to. So we could do it in another binding site for another AAV and say, hey, this AAV binds to heparin sulfate. Let's play around with that binding. Another way that we can do it is by actually, and this is something that the field is doing a lot of, is we can go into the AAV and try installing new peptides into those AAVs to give them novel properties. And we can do the same kind of thing where we mutate those peptides. Yes, I did. So here are this is the library screening results in both the mouse and the cyno. And actually, they do seem like they're doing okay in the muscle. So this could be something that could be worth looking at further in muscle tissues as well. So you can see, sorry, yes, let's talk about this. We've got, so everything here is normalized to AAV9. So AAV9 is going to be 1. So anything above this line means it's better than AAV9. And you can see that in the gastrocnemius of both the mouse and the non-human primate, we're seeing an enhancement over AAV9 for our XRH vectors. So yes. Mm-hmm. That's a great question. And that actually brings me to the other slide that I wanted to have just in case. So yeah, this is something that I think is really cool and really interesting. So what we've got here, we've got RNA over DNA ratio. So you take the number that you get for the RNA value and the number that you get from the DNA value, divide them by each other, and then normalize everything to AV9. So AV9 is 1. And what this is telling us is that especially in the heart, we're seeing this increased transcription off of our capsids that is not accompanied by increased biodistribution. So even though they're getting the same amount of vector in, we're getting more expression off of our XRH vectors compared to AAV9. And this is something that that's been around in the field for a little bit. If you look around at all the different, at a bunch of these different capsid engineering papers, kind of hidden in the supplementary figures, you will see evidence that these vectors at the DNA level are entering these cells, but at the RNA level, they have some type of enhanced expression. So what do I think? If you ask me to speculate on what is happening, I think that the capsid has interactions, and again, this is speculative, but my opinion is that the capsid is interacting with host cell proteins that allow for enhanced transcription once it gets inside of the cell after it's extruded its gene. So what that looks like, we don't really know, but that's another thing that I would love to study. Yeah. Thank you. So, and so this is my favorite observation from this data set. And I think that it depends on what you're trying to do. So you have an AAV, and it seems like for liver transduction, if we go back to our liver transduction, the thing that's being affected there is we're just getting less liver into the cell, or getting less vector into the liver. And there we're trying to reduce the amount of capsid, reduce the amount of expression that we're getting. Right? So if you want to make it go away, the best way is to just make sure that the vector doesn't get there in the first place. However, in the heart, we're seeing that these RNA over DNA ratios are much higher, which suggests that maybe the barrier to high cardiac transduction isn't necessarily getting the vector to the cell. There may be some other intracellular factors at play that are damping our ability to transduce well. So I think it depends. So I think it's hard to say what exactly a good expression ratio is, especially because it's going to depend wildly on the dose that we're using. I think you could see how too much expression could also be a problem if you are, overloading the cell's ability to transcribe other proteins. That could be a problem. So I don't know enough to say what the correct limit would be, unfortunately. But in general, more is probably going to be a little bit better until we get data that suggests otherwise. So. Mhm. So, and this is, so there's a couple things there. So a common tool for studying glycans in general. There are these two proteins. There are neuraminases, and so there's a protein called neuraminidase, and that cleaves sialic acid and exposes galactose. But then there are also this class of proteins called lectins, which are able to bind to these different proteins. So theoretically, if we wanted to block the galactose, we could maybe pre-inject with the galactose binding lectin and see if that has any effects. I haven't seen any evidence that that's been done yet, but if you pre-inject with this neuraminidase and expose more galactose, this is work that was done out of the Ahsokan lab, again, back in 2012. You find that you actually enhance the liver transducting that you see by exposing more. So yes, if we play around it, if we play around with the exposed galactose levels, we can definitely see. I've got some extended acknowledgements. Yeah. Yeah. Okay, so we're gonna X out of this. We're gonna stop sharing. Let's see if I can do technology. Okay, wait, I need to share my screen first. All right, so extended acknowledgements. So first, I would like to thank Dr. Wilson, Jim, for giving me the opportunity to come to the gene therapy program and to learn from the amazing group of scientists that I've had the privilege to be around and to absorb all the prodigious amounts of information that live in their brains. So thank you so much for giving me this opportunity. Next, I'd like to thank my thesis committee, Charlene, Denise, Vlad, Dan, and Dan, for just listening to me ramble about my research once every blue moon. It has, these conversations have given me a lot of insight into things that I could do better, and have really helped my development as a scientist. So thank you. I would also like to thank GTP broadly. There are so many people at GTP. I'm going to thank more people later. But in particular, Josh, Jenny, Dave, Rosie, Jeff, Jason, and Marcella, you senior scientists have really taken an interest in me and guided me through this process, and I really appreciate everything that you've done. Furthermore, GTP is this unique organization that has so many different core facilities and scientists with a number of different specialties that I am so lucky to have been able to work with and to enable my work. So the work that you just saw today could not have been done without the help of all these people. And so for that, thank you. Next, I need to thank Nathan Denton, who throughout the thesis writing and drafting and drafting and drafting and drafting process has been my constant companion. And this thesis would not have happened without his input. Next, the capsid team, Shiva, Sharon, Angela, and Payal. Thank you guys so much for being my lunch buddies. I really appreciate it. Also, I want to thank the Wilson Lab grad students. This is when we dressed up for Halloween as the AAV cereal types. No. Kalyani's was the best. AAV won bunches of oats. But a PhD in the Wilson Lab is like no other place to do your PhD. And so having a couple people that I could share that with really, really made a difference. Next, I want to thank my family, my mom and my dad and my brother Josh. I could not have done this without you. You were with me. I feel like everybody says this, but it's true. You were with me through the highs and the lows, picking me up when I fell down and just keeping me on the right path. And so words cannot express how grateful I am. So thank you. I also want to thank my wonderful cousins, some of which are here today. It's like having, another set of siblings around. To go through life with you all has been a privilege. Then my aunts and uncles, some of which are also here today. I love you all so much. And finally, JoJo, my grandmother, pictured here, safely observing the eclipse. So My grandmother's 90 years old, and she still has enough. And she has credibly made threats against my thesis committee. Now, what those threats are, I don't want to say, but you're warned. So finally, I want to talk about some of the people that I've lost during this time. This is my grandfather, and he unfortunately couldn't make it. And he promised that he would be here. And right before this presentation, my uncle Mark came up to me and he gave me this, which was my grandfather's, it was his Air Force coin that the pilots would take with them when they were going on a mission or doing something important. And so he kept his promise. I also want to talk about Abhinanda, who I wish could have been here today. Finally, my dog Opal, who continued to fight till the end. Next, I want to talk to GTV. Our culture and our togetherness is something truly unique, and I am honored to have been a part of it. That culture is a choice, and I implore you guys to continue to make it. To the people at Agnes in Touch, who I've been spending my Tuesday evenings with for the past year, getting to do something good every week has been just amazing. And so thank you for being a part of this. And finally, I just want to thank all of my friends. There are so many of you that have been a part of this journey for me, and I am so honored and privileged to have gotten to share this time with all of you. So here's Philadelphia. And then before we go, this is a great picture. So I'm going to do some sentimental stuff now. I've got a very short story that I'd like to share with you all. This is a story that I heard originally from my camp director, and I've adapted it since I first heard it. I'll share it with you. It's called The Most Perfect Heart. A young man stood in the middle of a town and proclaimed, "Come, look at my heart, for it is the most perfect heart in all the world." A large crowd gathered, admiring his heart, and they were in awe, for it was indeed perfect. There was not a mark or a flaw upon it. After some time, an old man pushed to the front of the crowd and said, "You are wrong. Your heart is not nearly as perfect as mine." And he opened his chest and he showed them his heart. The old man's heart was not perfect, not even close. It was full of ridges and mountains, valleys and deep holes. There were pieces missing and more than a few rough edges. The young man looked at the old man's heart with confusion. "You must be joking," he said. "Compare your heart with mine. Mine is perfect with not a scratch on it, and yours is a mess of scars and tears." "Yes," said the old man. "Yours is perfect looking, but mine is more beautiful. You see, every scar represents a person to whom I have given my love. I take out a piece of my heart and I give it to them. Often, they give me a piece of theirs in return. This fills in the empty space, but because the pieces aren't exact, there are ridges and valleys and a few rough edges. My heart, you see, is alive. Sometimes I will give a piece of my heart away and not receive a piece in return. These are the valleys, and within these valleys lives sadness. Where another has given of their heart with abundance, you find mountains, and upon those sunny peaks live joy, taking flight on wings of laughter. Other times, somebody that you love will pass, and in their passing, leave a hole so deep that only grief can live there. Giving of your heart is taking a chance. You know not whether you'll be left with a mountain or a valley, joy or sadness. The only certainty is that the more you give of it, the more alive it becomes." The young man stood in silence, tears running down his cheeks. He reached a hand into his perfect heart, and he took out a piece. He offered it to the old man. The old man took his offering, placed it in his heart, and gave him a piece in return. It fit, but not perfectly. There were a few rough edges. Everybody in this room, everybody listening on Zoom, everybody who has been here with me during this hugely significant period of my life, you all have a piece of my heart and a place of my heart. Thank you.
I want to. Jake will stay. During today and after. So.
All right. Thanks.